going to see central dogma and 3 types of RNA. So let us see what is central dogma. According to Francis Crick, genetic information always flows in one direction only. That is, we can say in unidirectional. How? Suppose this is DNA, and from the DNA, what is synthesized? RNA. And from RNA, we are going to form the protein chain. So, from DNA to RNA, when the information is transferred, is called transcription. And from mRNA, when protein chain is synthesized, is called translation. So, this is called central dogma. There were two scientists, Tamin and Baltimore. They have found one reverse transcriptase enzyme. We know that some of the viruses have genetic material or genetic information RNA instead of DNA. So in this case, how the transcription will take place? So this enzyme was used here. RNA was converted into DNA by reverse transcriptase enzyme. And now this from DNA, mRNA was synthesized, which we are going to say transcription. And from mRNA, protein chain will be synthesized, we are going to say translation. In short, we can see here, this is RNA, the genetic material of virus, which is converted into DNA by the enzyme. From the DNA, again it is converted into mRNA, and then mRNA changes or forms protein. So here the information is flowing in two directions, so it is bidirection. This is called central dogma reverse. Now let us see 3 types of RNA. The first is mRNA, known as messenger RNA. The structure of this already we have seen. Just I will like to explain that it is having capping of, if you remember, MG-PP, and tailing is by poly A tail. So initiation codon is present AUG, and stop codons are present. These are the 3 stop codons on the mRNA. Now the second type is known as tRNA. T stands for transfer. The structure of this tRNA is Cloverleaf, so it is called Cloverleaf model of tRNA. Here they are having 4 arms and 1 small arm, the 5th. The first arm on the top is having the code CCA on 3' direction on hydroxyl group. Other one is 5N, which is having the nitrogen base guanosine. This particular arm is called acceptor arm because here amino acid is going to attach. So it is attachment site for amino acid. Now on left hand side, this is called DHU arm, which is known as dihydroxyuridine. Here enzyme recognition site it is called, and here specific amino acid activating enzyme is present. Then we move to the right side arm. This is called ribothiamidine T, it stands for pseudouridine, this is the symbol of it. Ribocytidine is C. This arm, actually here ribosome is going to attach. Then this arm opposite to the acceptor arm is called anticodon arm, where anticode means on the mRNA the code which is brought AUG, suppose this is initiation codon, what will be the anticodon of this will be A matches to T, but it will be U, and U with the A and G for the C. So this anticodon is brought by this tRNA for every genetic code present on mRNA. In short, we can say anticodon reads its appropriate codon on mRNA and temporarily binds to it. So this is called recognition and also. Now let us see the structure of rRNA. R stands for ribosomal RNA, which is present 80% in a cell. This rRNA is made up of two subunits, large subunit that is 60S and small subunit of 40S. Now see the structure of large subunit. This is the large subunit. Inside it there are three sites. On the right hand side is A site, which is where the tRNA, new tRNA is going to join, or it is called acceptor site or amino acyl site. Then P stands for peptidyl donor site. Here actually in the starting, initiator tRNA joins in the starting of protein synthesis. Then there is one more site called E site, which stands for exit site, which is on the left hand side of P, through which the tRNA will be moving out. Now let us see the structure of small subunit of ribosome. This is small subunit of ribosome, which is stuck on the mRNA, or which is joined on the mRNA, and which will move on the mRNA on the right side to synthesize the polypeptide chain brought by the tRNAs. So at last, this small subunit of ribosome is helping to synthesize the protein chain. Let us see now conclusion of this. In every cell there are 20 amino acids present. So there should be 20 or more types of tRNAs, and along with that 20 enzymes to activate this amino acid should be present in the cell. That's all for today.